Hello everyone. This video is in continuation to our videos for grade 12 molecular basis of inheritance. In last video we had discussed the structure of RNA. In this video we'll start with the first division of RNA, that is messenger RNA or mRNA. Messenger RNA, I'll be talking about how it is formed, then we'll in detail talk about the structure of mRNA. So watch the video till the end and please share, subscribe and like the channel. Share it with your seniors and juniors. Grade 10th videos are also made, 11th and 12th are also made for biology. Let's start. mRNA, messenger RNA. We will first talk about the two people. There are two people who gave this idea of having an mRNA. RNA तो था पहले से. That is Jacob and Monod. Jacob and Monod gave this idea in 1960 that there should be or there must be an mRNA which is responsible for bringing information from DNA for protein synthesis to RNA, to ribosomes of course. Fine. So this idea was given by Jacob and Monod. Now RNA is formed on DNA, as in it is formed on a DNA strand and then spliced off. So RNA is formed on DNA. Now what is this process known as? The process by which it is formed is known as transcription. So if someone, or if you have to have a clarity in your head, formation of RNA, the process of formation of RNA is transcription. We'll see the steps of that synthesis in detail. So let's see how it is, basically an idea how it is formed. Suppose this is a DNA molecule, or sorry, DNA strand. If we split open a DNA, so it's a double stranded structure. So we have split open. So suppose this is one strand of DNA and suppose we write a hypothetical code over it like A, T, C, G, A, T, G, C. Suppose, and when an RNA is formed over it, and of course we have the 3 and 5 prime states. When it is formed over it, of course the antiparallel purines always bind with pyrimidines. The same way adenine with uracil. Why uracil? I'll just use another color. Why uracil? Because in RNA, as we discussed in the last video, thiamine is converted, or you can say replaced by uracil. So adenine with uracil, thiamine of course with adenine, cytosine with guanine, guanine with cytosine, A again with U, T with A, G with C and C with G. So this is how our RNA strand is formed. This is how or what we get when an RNA strand is formed. Now let's see the steps of transcription. Steps of transcription. Now the first step, that there is a DNA strand. So there is a DNA strand which further, now this whole DNA strand is going to bifurcate, or it will be divided into two parts, introns and exons. I'll just shade the introns, and exons I leave unshaded. So this shaded part, I'll tell you what it is. Introns, this is exon, then intron, then exon, intron and exon. So suppose this is E1, E2, E3. It is I1, I2 and I3. Now this structure which we have got here is a heteronuclear RNA, hRNA. It is a heteronuclear RNA. Now it has two parts, introns which are non-coding. They don't code for anything, but they are not required in RNA. And then there are exons which are coding parts, the ones which are coded structures which we need for the process of formation, or which we need in an RNA. So introns and exons are there. This is a heteronuclear. So what is the first step when the heteronuclear RNA which we receive when the DNA divides into introns and exons, that is a carrier of non-coding and coding parts, heteronuclear RNA. And then what happens with the process or by the usage of enzymes, splicing takes place. Splicing means slicing with the enzyme endonuclease. This endonuclease enzyme will split up the introns and exons. How many did we have? I1, E1, I2, E2 and I3, and we also had E3. So shaded part, shaded part, shaded part. So there is a splitting which is taking place, and which enzyme is responsible for that? Endonuclease enzyme is responsible for that. Now we don't need introns, so introns are left, and what happens now? The exons, they combine together. How? E1, E2, E3. They combine together, and this is done with the help of enzyme ligase. Ligase उनको glue कर देता है, दोबारा attach कर देता है, and then this unstable RNA molecule is formed. And how is it made stable? This unstable RNA molecule is now made stable by attaching two methylated caps on both the sides, the 5' and 3' end. Now this, the resultant which we are going to get is a stable RNA molecule. Fine. So I'm just quickly going to repeat it. First step is, as I told you, we get it from DNA. How is this? I've already explained कि ये कैसे changes आते हैं. Herein, a complementary strand is RNA strand is formed. अब herein we have introns and exons. Introns are the non-coding parts and exons are the coding parts. We don't need introns in RNA. So they are, first of all, everything is spliced up or divided by using the enzyme endonuclease. Then introns is not required, so it's gone off, and then exons, they join off to form an unstable RNA molecule, which further is made stable by attachment of two methylated cap on both the sides of RNA. So this is the process of transcription. Now we are going to discuss the structure of RNA. Fine. So let's see the structure of RNA. I've already made the structure here. Let's see the structure here. Let's make the structure. So what do we have here? We have, again, how did we start? We have a cap here and the whole strand. Again a cap here. Let's see how it is divided and what all parts are there in it. So when we see the structure herein, this is the 5' end, 5' and 3' end. At 5' end, there is a, I'll just demarcate this. At 5' end, there is a cap which is methylated cap is present at the 5' end. And what is it? Methylated guanine. Cap is present at the 5' end. 3' end pe also there is a cap. It is a polyadenine tail. Polyadenine, why do we call it? Multiple adenine molecules, they form a chain here. After this, in the structure of genes, we have a start codon, a stop codon. There is one start codon and that is always AUG, and again there is a stop codon here, stop codon. There are three, UAG, UAA, UGA. Fine. So any of these, तीनों इकट्ठे नहीं, any of these can be present. So one stop codon, and this is the structure of gene. So this is how an mRNA molecule is. I'm again repeating. mRNA has 5' and 3' end. 5' ends. This is specific kind change. 5' end, a methylated guanine cap is there. starts with a start codon AUG, stops with a stop codon any one of the three. There is a structure of gene in between, that is the sequence. And then at 3' there is a polyadenine chain present. I'm not going to write all this because the structure is self-explanatory. What I'm going to write here is that mRNA is a short-lived structure. Once the protein for which it codes is formed, this dissociates and a new mRNA is formed for proteins. I'll just explain in Hindi, थोड़ा सा confusing है. mRNA इतना शॉर्ट लिव्ड है कि जिस प्रोटीन के लिए इसने कोड करना है, अगर वो प्रोटीन एक बार बन गया, सपोज वही प्रोटीन दोबारा बनना है तो उसी mRNA से नहीं बन सकता. A new fresh mRNA is formed and a new fresh protein, be it the same protein, is formed. So that is why we consider it a short-lived structure. So in this video, I've explained you mRNA, the process of transcription and the structure of RNA. In our next video, we'll take up the structure of tRNA. So keep watching, liking, sharing, subscribing. Thank you.